All right, good morning or afternoon or whatever it might be. In class, we just finished talking about the structure and function of microbes. And now we're going to move on and we're going to take that structure and we're going to start talking more about the function. And specifically, we're going to talk about the function of DNA and RNA and its role in transcription and translation. And yeah, I know you're going to hear, oh my gosh, I've heard this before. Another lecture on the central dogma. I'm hoping this one's a little bit more detailed and it's going to take it in surprising directions and show you some of the differences between bacteria and archaea, because those differences are significant. All right, so first of all, let's look at the chemistry of RNA and DNA. There's two major differences. RNA has a 2' hydroxyl, DNA does not. And RNA uses uracil, where DNA uses thymine. Why? Well, the 2' hydroxyl is actually catalytic, and it can be autocatalytic and cause the cleavage of the nucleic acid. And this will happen in RNA, and it's actually part of the splicing mechanism of RNA. The other thing is that that 2' hydroxyl, the lack of it makes DNA have a wider, shallower major groove so that proteins have better access, and the DNA is less rigid, so it makes it easier to wrap around. So that's why you don't have the 2' hydroxyl in DNA. Why thymine? Okay, this one's a little bit convoluted, but in RNA and DNA, cytosine can degrade to uracil. If that happens in an RNA molecule, you don't know if that uracil is correct or not because there's uracil in the RNA molecule. If that degradation of cytosine to uracil happens in a DNA molecule, you know that uracil is not supposed to be there, and you can remove it. So it's a way of making sure that you don't get these degradations. Now you might think, wait a minute, isn't this a problem for RNA? And in fact, it's not. The half-life of DNA is on the order of days or weeks or months or years. Right? You want to keep that for a long, long time. The necessary half-life of RNA is in the realm of minutes or hours or maybe a day. So RNA is much shorter lived, and you don't, in its time frame, its lifespan, this really isn't a problem with the cytosines going to uracil. What we're really saying is function dictates structure. Since DNA needs to do these things, it needs to be more stable, it needs to be packed, you don't have a 2' hydroxyl, and you use thymine instead of uracil. Also, DNA is double-stranded. Why? It's a useful way to store information because you can copy the strands. It's more stable. It's easier to package a double strand. And this flexibility of the DNA from the lack of 2' hydroxyls makes it much easier to wrap around histones. RNA is single or double-stranded. It's used in temporary messages like messenger RNA, in enzymatic reactions like the ribosome, and a translator and adapter in tRNA. So you also have ribosomes and riboswitches, and we'll cover those in another lecture. The DNA has double-stranded reasons. What holds that DNA together? Well, as far as how it hybridizes together, the A's pair with T's and the G's pair with C's, as you know. And these are pretty reasonably stable structures. That base pairing contributes significantly to it, but the really big thing is base stacking, as you see here on the right. Base stacking is very important. These hydrophobic interactions, these van der Waals forces in between these DNA molecules, and this association makes this a very attractive structure, and that is actually what stabilizes DNA. The other thing to know is there's a directionality of the DNA. Remember, the DNA strand is anti-parallel, right? It goes 5' to 3' on one strand and 5' to 3' on the other strand. There's a directionality in how RNA is transcribed. RNA is recognized as a promoter, as we'll talk about in a second, and is transcribed from the 5' to 3' direction. As I said, RNA will have secondary structure. Yes, lots of it is single-stranded, but that single strand will wrap along itself and hybridize, and here's a couple examples. Transfer RNA will form these loops, and it's this L shape as shown here, and these various loops, and these loops are important. The anticodon region is one region, the acceptor end where the amino acid binds is another. The 16S ribosomal RNA and the 5S ribosomal RNA also have very specific secondary structures and will hybridize in specific ways. DNA size. Genomes of various organisms can be of many different sizes. The smallest known genome is Carcinella ruddii. It's 160,000 base pairs, and that's pretty amazing because there's actually some plasmids that are larger than that. This is an intracellular endosymbiont of sap-sucking insects, and we'll talk about that, and what they do is provide amino acids for the insect. The average bacterium, E. coli, is about 4.6 million base pairs, about 4,300 genes. And the largest genome ever measured is Amoeba dubia, which is 684 billion base pairs. Now, that huge amount of base pairs only codes for 50,000 genes, so there's obviously a lot of non-coding regions in there. But clearly what you should get from this is there's no correlation with genome size and organism complexity. Humans, if you're curious, are about 3 billion base pairs, or about 21,000 genes. Let's talk about the central dogma. It's a blueprint. As you know, a blueprint is needed to make proteins. This in turn makes new copies of the blueprint, and that's the whole deal. DNA is transcribed into messenger RNA, that's translated into proteins, and then these proteins carry out the function of the cell. One of them is to make more DNA. Howard Temin, who is a professor at this university, and David Baltimore actually modified this a bit by finding proteins that could reverse transcribe messenger RNA into DNA, and these proteins were coded in viruses, and the most famous one is probably the HIV virus. But the general flow is still from DNA to RNA to protein. All right, so what is a gene? A gene is a region of the chromosome that's a discrete unit of hereditary information. It's a segment of DNA that specifies a protein or proteins or an RNA molecule that then carries out some function. Now, microbial genomes are mostly genes. They don't have very little non-coding sequence. Here you see it. You've got a promoter here. You've got a gene right here. And this is what's called a monocistronic RNA. It only codes for one gene. It's not uncommon in bacteria to have what are called polycistronic RNAs. That is where you have a promoter and a series of genes that are transcribed all in the same messenger RNA. How does a DNA sequence direct protein synthesis? If you just look at a bunch of DNA sequence, and here's an example from mycoplasma, it's the long strings of A's and T's and G's and C's. But how does a cell know where to make RNA? Where to translate proteins? It turns out there are proteins that recognize sequences of base pairs, and these sequences flag start sites, stop sites, translation start, translation stop sites. There has to be enough complexity so it doesn't appear randomly by chance. Here we have an example of a promoter. Here is a start codon. Here is a stop codon. This is the protein coding sequence in here, and then you have transcription termination. The first step in moving from DNA to protein we will start with is transcription, and that is copying DNA into RNA. Here's a nice little graphics of what it looks like. So some general thoughts and things to consider. Transcription is performed by RNA polymerase. This is probably one of the more conserved enzymes in nature. It identifies genes by promoters. In bacteria, there's a -10 region and a -35 region, and the -10 means it's upstream 10 base pairs from where transcription starts. The promoter is recognized by a sigma subunit of RNA polymerase. Promoter strength can be different, and the strength is determined by how well the sigma factor, which is one of the proteins in RNA polymerase, recognizes it. This is in turn dependent upon how close the sequence is to an ideal sequence or consensus sequence. The closer that is, the more tightly sigma can bind, the more likely you're going to get binding and transcription. Now, different promoters can also be recognized by different sigma factors. There's not just one, there's a whole bunch of different ones. And this can be a major point of regulation, as we'll find out later on in the semester. So here's RNA polymerase. This is an RNA polymerase, and I use a molecular graphics program to actually make a little molecular model of it. And here's the two alpha subunits. Here's a omega subunit. Here is beta subunit. Here's the beta prime subunit. And then here is the RNA in purple here, and the DNA in red. And the sigma factor is not shown on his here because this isn't an elongation complex. So the steps in transcription, you get recognition, binding to promoter, you get formation of the open complex, so this spreads open here. You then get elongation, it moves along the DNA and transcribes it, and then finally termination. And then you can see the bases going in here. Recognition in the bacteria, the sigma subunit, as shown here. Here's a little cartoon now of RNA polymerase. The sigma subunit is in here. It recognizes the special sequence, as we talked about. This is also called the Pribnow box. RNA polymerase then binds, transitions to open complex, opens about 12 base pairs, and then you move on to elongation. The RNA polymerase makes a new strand, and it moves in the 5' to 3' direction. So this is going 5' to 3'. Here is the DNA, and here's the RNA coming out of channel. As it moves down the DNA, random nucleotides enter the channel. So the DNTPs come in here in this channel, and it's just random nucleotides until one binds and it fits, and then it goes on to the next one. The rate is 45 base pairs per second, and this moves at the same speed as translation, and you'll find out why that's important later on in the lecture. After about 20 nucleotides, RNA starts to emerge from the polymerase. The sigma may fall off at this point, or it may be retained. So which strand of the DNA is made into RNA? Right? There's two strands of DNA. One serves as the template for the messenger RNA. That's shown here. The other serves as a coding strand. So the template is the one that's copied. The coding strand is what the messenger RNA looks like when it's done. Right? So the other is the coding strand, except you have T's instead of U's. In genes, it can be either strand. It's not like all one in the genome, one strand is a coding strand and one strand is a template strand. It can switch back and forth. You then go on to termination. You recognize a termination signal and it releases the complex. And this can happen in two ways. One is row-dependent termination. So what happens is that the RNA polymerase gets here, gets past the last gene, and you start to have this area of open RNA that doesn't have ribosomes on it. And then the row will bind to this open sequence, hydrolyze ATP, move towards the polymerase, and when it gets there, it will yank the RNA out from the RNA polymerase. The other one is row-independent termination. And what happens is, at the end of the sequence, there is this inverted repeat. And this inverted repeat can then form a hybridization. It can form a structure. So hybridize together, and this causes tension. And then there's a run of U's after it, right? And they don't bind very well because it's a little bit weaker binding. And this tension here will just pop it right out and it falls off. As we talked about, prokaryotic messenger RNAs contain multiple genes. They're polycistronic. Often, operons that are involved in regulating some function will code for their genes in these polycistronic messenger RNAs. So this is a very common motif that you'll see in bacteria. One example is the degradation of lactose, and here is the setup of the operon. You have a promoter, ZYA, the three genes, and a terminator, and they're all transcribed together. There are differences across the domains. If you look at bacteria versus archaea, they will both have a single RNA polymerase, but the RNA polymerase from archaea has a different structure. In fact, that structure is very similar to eukarya. The promoters are different. The promoter structure seen in archaea is again very similar or somewhat similar to what's seen in eukaryotes. However, the messenger RNAs that are made are fairly similar. They're often polycistronic and they're both relatively unstable. If you compare bacteria versus eukaryotes, eukaryotes first of all have a number of different types of RNA polymerases. The promoters are different, and again, eukarya and archaea will have similar promoters structures. And the mRNAs are different. Eukaryotes, because they have a nucleus, and this messenger RNA has to be made in the nucleus and then move out into the cytoplasm, they will make them monocistronic. They will put a cap on them, they will put a tail on them, and that makes them much more stable, or more relatively stable than the messenger RNAs from bacteria and archaea. Eukaryota and archaea have introns. They will have intervening sequences. Almost always bacteria do not. If you look at the RNA polymerases from bacteria and archaea and eukarya, you see a difference. The bacterial RNA polymerase is different, right, than the archaeal RNA polymerase. And the archaeal RNA polymerase looks very similar to the eukaryotic one. As I said, there are also differences in the promoters. Archaeal and eukaryotic promoters are different than bacterial promoters. Instead of this -10 and -35 region, you just have this ta-ta box that is recognized. It's only 4 base pairs long. And obviously there's more signals that must be important for recognizing a promoter. And there are. There's an initiation sequence, there's this Brie sequence, etc., that recognizes it as a promoter. The ta-ta binding protein, or TBP, will recognize the region first, shown here, and then the transcription factor B will bind, and then that recruits the RNA polymerase, and then you get transcription. In eukarya, it's a similar method, except the proteins that are involved are a little bit more complex, and we're not going to go into the details. So, for processing, archaea and eukarya have these introns, and these introns need to be spliced out. So you get these endonuclease cleavages, and then you get a ligation and a mature spliced tRNA. Same process happens in eukaryotes. And in fact, not only do they have the introns spliced out, you also get a cap and a poly A tail put on, and then that makes a mature messenger RNA and the protein. Okay, that's what I wanted to say about transcription. Let's now move on to translation. So in this case, we're moving from nucleotides to amino acids. And what you want to really understand is, how do you go from the code to a specific sequence of amino acids like met, serine, pro, or whatever. So you need a few components. You need a translation code, some way of knowing what to translate to one. You need an adapter that recognizes both nucleotides and amino acids. And then you need a machine to make the peptide bond. Translation is the most conserved process in biology. It was probably the first function that evolved in a cell, and involves the converting of nucleic acid sequence to amino acid sequence, and it's a new language. A few points to remember about the genetic code. The genetic code is made up of triplets, and these triplets dictate which amino acid gets put in. The real translator or adapter molecule is tRNA. The genetic code is universal. This is used in all organisms. Also notice there is redundancy in the code. More than one triplet may code for the same amino acid, and you'll also notice, very interestingly, these redundancies are always in the third base pair of the codon. And this wobble, as it's called, is very clever because it prevents mutations, and we'll go into that when we talk about mutations and replication. The real translation occurs not at the ribosome. That is just matching two different nucleic acids, right? A tRNA to an messenger RNA. The real translation happens when the tRNA is charged by the tRNA synthetase. So you have to charge with the correct amino acid, and it's got to happen at the right time and the right amounts. So why is this so important? Because if you don't do it right, you'll get random amino acids put in, and that's a huge deal. The real process of translation takes place at these tRNA synthetases, and that's something to remember. So how does protein synthesis happen? I'll go through this really quickly because you've had this before. The 30S subunit plus a messenger RNA plus FMET tRNA plus initiation factor get together with the messenger RNA. Right? The messenger RNA is a 16S ribosomal RNA. It's the small subunit, and that binds and sets up the reading frame, and then FMET tRNA enters the P site. The 50S ribosome is added, and a second charged tRNA then enters the A site with help from an elongation factor. So here's the initiation complex. It binds and enters the P site. Then you have another one, enters the A site, and now you're ready for the next step, which is elongation, right? Peptide bonds form between the two. It's located at the tRNA A site. The ribosome advances and it moves everything over, right? So now the initially charged amino FMET tRNA is now in the E site, or exit site. The P site now has this tRNA with the 2 amino acids on it, and another amino acid then comes in. And by the way, this recognition is again random. Random tRNAs come in here and bind to these sites. So you have to try up to 20 different ones before one finally recognizes it, and boom, it goes in. So the newly charged tRNA enters the A site, peptide bond formation continues, and during every translocation, GTP is hydrolyzed to move this along. Eventually, you terminate a stop codon and release factors bind, and they pull the protein out. Not all of the mRNA codes for protein. There are sequences before the first methionine, the start codon, and there are sequences after. Right? This gets translated into a protein. You have the amino terminus, which is at the beginning of the protein. You have the carboxy terminus, which is at the end. It's called the amino terminus because there's an amino group on here, right? And all of these are linked together, so they don't have the amino groups. And at the end, you have the carboxy terminus because it will have a carboxyl group at the end. So what are these other things doing that aren't coding for protein? Well, you have the start region, the ribosome binding site, right? And then the AUG. You have a stop codon, which does not code for amino acid. And establishing that and getting the frame is absolutely critical to making a protein. One thing that's important to remember, transcription and translation in bacteria is coupled. This is very tightly coupled. And as soon as an RNA is made by RNA polymerase, ribosomes will begin to bind to it, and they will start translating it into proteins. And this happens very quickly. And this is one of the reasons that bacteria can respond so quickly to changes. They make their proteins very quickly. So this will go on, et cetera, et cetera. When the RNA polymerase gets to the terminator, these will go through, they'll finish the messenger RNA, and then they'll stop. Now, there's a very important consequence to this. Imagine that there's a mutation in this B protein such that it cannot continue, and there's a stop codon right in the middle of this B protein. What happens is the stop codon causes the ribosome to fall off, and obviously you don't make a functional B protein. But you also don't make the C and D proteins because you have this open space of messenger RNA. Remember that the Rho protein, which causes transcription termination, binds to open spaces of messenger RNA. Normally when a gene is transcribed and then translated, that open space doesn't occur till the end when it's past this D gene, and then you run into the terminator, so there's an open space there and it pulls it out. But now you've got an open space in the middle of the gene because of the stop codon in protein B. The Rho protein can bind and it will stop transcription, and this mutation here, the stop codon, will prevent the expression of not only B but also C and D. And these are called polar mutations. Okay, so what's the product of translation? Strings of amino acids, and these are functional products. This may require protein folding. What we used to think for a long time is that these proteins just spontaneously fold up, and some of them do. But there is a whole class of proteins called molecular chaperones, such as GRO-EL and GRO-ES, that are essential for the folding of many different proteins. So many proteins that are produced will be folded in GRO-ES, GRO-EL to make a properly folded active protein, and this is a very important function in bacterial cells. Also, if a improperly folded protein comes out due to heat or whatever, DNA K and DNA J, with the help of ATP, can also fold proteins properly. It just depends on the pathway in the protein. Okay, so what are the differences in eukaryotes? First of all, the mRNA is processed and then goes out to the ribosome, so it does not occur simultaneously. You do not get polycistronic messenger RNAs, and you do not get this coupling of transcription and translation. Translation initiation, there's no Shine-Dalgarno sequence. It will just start from the first AUG. And methionine is used, not F-methionine. So formal methionine is only found in bacteria, in fact. And here's a nice comparison chart of or the differences between the three domains in how transcription and translation occurs. And these are important distinctions, and I will bring them up in examples as we go through things when we look at how antibiotics work, why a drug works against bacteria, not eukarya, and vice versa, how the immune system recognizes bacterial proteins, etc., etc. But I'll save that for future lectures. Now, before I finish, I want to tell you a few interesting topics that they don't normally discuss when we talk about central dogma. Is that there's all sorts of exceptions and lots of examples in prokaryotes and in eukaryotes. GUG and UUG can sometimes serve as start codons. It just depends. The Shine-Dalgarno sequence is not always necessary. There are examples of genes that don't have Shine-Dalgarno sequences that get transcribed and translated just fine. Some proteins can have two different start sites, and you'll see this a lot in viruses. And then there's even examples of this. So here's an example of a protein, and what will happen is the tRNA will slide. You're just going along on a reading frame, it gets to here, and then it will just skip 2 bases and then go on again. Why? Who knows, but it works. Some other interesting exceptions. Selenocysteine, used in some proteins, and it's added at a UGA codon, is found both in bacteria and eukarya. If you think about it, the UGA codon is a stop codon. And yet in some instances, for example in selenocysteine and pyrrolysine, this stop codon is ignored. And what happens is there's a selenocysteine incorporation sequence. The context of the messenger RNA in bacteria after that UGA tells the translation machinery, no, this isn't a stop codon. You want to put selenocysteine in there. Now, example of protein found in this is mammalian thioredoxin reductase. That protein is a selenocysteine containing protein. Another example is the 22nd amino acid, pyrrolysine. It inserts at UAG, and this is found in some methanogens. An example is methanosarsina barkeri. It's monomethylamine methyltransferase proteins, and ones involved in methanogenesis are actually have pyrrolysine in them. And you can also get translation read-through. So in other cases, you'll just put in a trip codon, which is UGG normally, and you'll get a read-through, and again, you'll get 2 different proteins. So there's all sorts of interesting exceptions. Okay, that's the end of this. I'll see you in class where we'll actually do a number of exercises working with the central dogma to help you understand it at a deeper level.